All right, so this is the start of category 2 of the content, and now we'll be talking about molecules, cells, and organelles, essentially. So last week we talked about lipid metabolism, but we haven't really talked about what lipids are. Like we have talked about carbohydrates and polysaccharides and monosaccharides. We've talked about amino acids building up into proteins. And now we've introduced this idea of lipids. So what exactly are lipids and what defines a lipid, right? So to compare these things, a carbohydrate has a very specific ratio, 1 to 2 to 1 of carbon, hydrogen, and oxygen. So it looks like CHO. And those are the things like glucose and sucrose, the sugars that we saw, right? Amino acids and proteins. Proteins are made up of those amino acids, and amino acids have that N terminus and that C terminus that we talked about, right? The distinctive amine and carboxyl groups. Now, lipids, the major characteristic of lipids is that you'll see that they also have carbons, they also have oxygen, they also have hydrogen, right? So what differentiates a carbohydrate from a lipid? Well, the main thing is that they are hydrophobic. Hydrophobicity is what makes a lipid, okay? So here we have two different lipids, right? We have one type of lipid is right here is testosterone, right? Which you may be familiar with. And then here we have a triglyceride, like we saw last week, the things that we break down for energy, right? So these two things have different structures. And remember, structure informs function, right? So the bottom one, the triglyceride, we break down for energy. The top one is not suited for energy breakdown. What is it? It's actually a signaling molecule. So again, the structure informs the function, right? They look very different, but they're both hydrophobic lipids, right? The hydrocarbon ring structure of the testosterone and the hydrocarbon tail of the triglyceride causes these molecules to be nonpolar, right? So a long carbon chain or a big carbon ring is nonpolar because we showed the reasoning for why that is the solvation shell, right? So we talked about how if a hydrophobic molecule is in the water molecules around it, the aqueous liquid around it, has a lot of freedom to move around and interact with the molecules. However, if a hydrophobic molecule is in the water molecule has less freedom because it can't interact with that hydrophobic molecule, so that increases the rigidity of the water around it, right? So this is why hydrophobic molecules are like long carbon chains are hydrophobic because the water around it can't interact with it as well as it could with something hydrophilic. So they will be insoluble in polar solvents, right? What does that actually mean? So insoluble in polar solvents, we've all seen that thing where like oil and water don't mix, right? If you put oil and water together, they form separate layers, right? That's what insolubility means. If something is soluble, like for example, if you put sugar or salt in water, right, and you mix it, it will dissolve, right? And it will become one homogeneous mixture, right? That's solubility, right? Because the sugars have polarity and can interact with the water well. But these long lipid chains don't have polarity, they're nonpolar, they can't interact with the water well, and it's more favorable for them to just stay separate entirely. Okay? So that's the big deal with lipids. They are nonpolar. So because they are nonpolar, right? This gives them a lot of properties. This gives them a lot of useful factors in our biology. So one easy example is wax, right? If you look at a tree or a leaf and you feel like the leaf, sometimes they feel a little waxy. They feel a little you can just feel like they have a little glossy sheen to them, right? And that's actually waterproofing. That's waterproofing. That's one way because they're nonpolar, they repel the any water that comes in. So that's one form of lipid use in biology as a wax. Now, a major form is for signaling, right? We showed that triglycerides can be broken down for energy. Waxes can be protective. Signaling lipids are specialized lipids for signal transduction pathways, right? Which is passing information, right? We talked a little bit about signaling with like G proteins and stuff. Actually, this might be, it might be close to here. Yeah. So, oh yeah, this is pretty close to it. Yes. So for example, signaling, so the signaling lipids are lipids that are just bind and cause a signal communication to happen, right? So these lipids are in two categories, steroids and fat-soluble vitamins. So steroids are a four-ring structure like this, which has 3 cyclohexanes, 6 rings, 6 rings, 6 rings. That's a hexane, and a pentane, a 5-ring, right? So these hydrocarbon rings make steroids nonpolar. And cholesterol is the major steroid that you should know about. Why? Because it's a huge part of the membrane, and we're going to talk about that. So steroid hormones are secreted by endocrine glands, and they act as hormones, and we're going to talk about that more when we cover, anatomy and systems and the endocrine system. But this is basically the template of what a steroid looks like. Now, testosterone, cholesterol, there's a lot of different ones. They all have this like 4-ring kind of structure. Now, terpenes are a different kind. We can look at, actually, before we talk about terpenes, since I have prostaglandins here, let's talk about prostaglandins. So prostaglandins are a class of lipid hormone. Unlike steroid hormones, the steroid hormones are like based on cholesterol. Prostaglandins are based on arachidonic acid, right? And that just means that they look like this, and they have a 5-carbon ring. And what do prostaglandins do? They basically act as signaling molecules. Autocrine and paracrine means, autocrine means acting on the self. So whatever cell secreted it, it will act on its own cell. Paracrine means juxtacrine is right nearby. Paracrine means like around. I think it's, let me check. Let me just check. Juxtacrine is right next to it. So paracrine, paracrine. Nearby, yes, paracrine is nearby. This is an aside. This is more towards endocrine system, but this is just an aside. Autocrine is on itself, paracrine is nearby, juxtacrine is directly next to, and endocrine is into the bloodstream, so it can go throughout the cell. That's just an aside. So back to prostaglandins. The main thing that prostaglandins do are basically like helping with inflammation. So the arachidonic acid can get turned into a prostaglandin, and the prostaglandin comes and attaches to a receptor, and then it induces inflammation in the cell. One major thing to note about lipid signaling molecules is that lipid signaling molecules are very non-polar, right? So as non-polar molecules, they don't go inside the cell, because inside the cell is an aqueous environment, right? So rather, it makes more sense for them to stay at the surface and attach to something at the surface. And when they do that, inside the cell, they can trigger a chain of signaling that happens inside the cell. But they themselves do not enter the cell directly. That's an important thing to understand, because it contrasts with other types of signaling molecules, which may be small and polar and go through into the cell. Okay, prostaglandins. And then terpenes are just a different kind. Terpenes are involved in the synthesis of cholesterol. So this is called an isoprene. This is just a type of hydrocarbon chain. And when isoprenes build up, they turn into cholesterol, essentially. That's essentially how cholesterol is made, with this subunit building up at a time. So this is just some vocabulary that an isoprene is this. A terpene refers to 2 isoprenes. So one terpene is 2 isoprenes. And then a squalene is 3 terpenes, but since each terpene is 2 isoprenes, then it's 6 isoprenes. And then a cholesterol is like this stuff just rearranged into that 4-ring template that we talked about. So that's just the most thing that you would need to know about terpenes is that they are involved in building up cholesterol and the relevant hormones that are offshoots of cholesterol, like vitamin A or like testosterone. So that's terpenes, prostaglandins. Then vitamins. So there are two different types of vitamins: water soluble and fat soluble, right? So if they're fat soluble, then they're lipids. So and so the 4 fat soluble vitamins are A, D, E, K, right? And the rest of them are just water soluble, right? So the fat soluble vitamins will stay in the adipose tissue while the water soluble vitamins don't. And overall, right? Overall, so what do we say lipids are? Lipids are insoluble in water. That's the major thing. And so likewise, they would be soluble in an organic solvent that's nonpolar, right? Some kind of nonpolar liquid. So the monomers of a lipid, right, are glycerol and fatty acids, as we saw with, well, that's specifically for like triglycerides, right? Whereas for the other one, like we saw, it's like terpene is the monomer, right? They're hydrophobic, and there's hydrolyzable lipids and non-hydrolyzable lipids. So what does that mean? So hydrolyzable lipids can be broken down. So if you look on the right here, hydrolyzable lipids, like for example, with this triglyceride, right? We talked about lipid metabolism. We talked about breaking these molecules down. How do they get broken down? There's a glycerol backbone, and then there's these long carbon chains, fatty acids, right? Hydrolyzable means that at the red here, at the red bond right there, let me just draw it out. Where is my... Okay. So at the red bond here, right? This can be hydrolyzed. So what that means is a molecule of water can enter here, and the water will get split as an H here and an OH over here. So that's going to give us a carboxylic acid at the end here and a hydroxyl over here. So this is hydrolysis, right? We can break this. Not all lipids can be broken like this. You have to have this kind of, carbonyl bond to be broken. If you see that kind of pattern, then the lipid is hydrolyzable. However, there's lipids that are not hydrolyzable, like the ones with the carbon rings, right? So that's a big difference. So the hydrolyzable lipids are triglycerides, phospholipids, and waxes, whereas the non-hydrolyzable ones are steroids, like those big rings, the prostaglandins, and the vitamins, right? So if we look back at the prostaglandin, we'll see that they don't have that kind of carbonyl attached to a leaving group that we can break. Yes, there are carbonyls here, but this carbonyl is only attached to carbons, so you can't break those bonds. And this carboxylic acid is already a carboxylic acid, right? There's nothing more to hydrolyze here. So these are the major types of lipids that we see. Those are the major types of signaling lipids that we see. Now we move on to structural lipids. And structural lipids, again, lipids are incredibly important because of their nonpolar property. So that nonpolar, we can utilize that. That's useful to us. There's also lipids by themselves are hydrophobic, right? They have long carbon chains. However, there are some lipids that also have hydrophilic parts to them. So remember, hydrophobic means that they are not interacting well with the water. Hydrophilic means they are interacting well with the water. They like to be with the water, right? That's called amphipathic. You might have heard of like ambidextrous, right? Meaning like you can write with both hands. In this case, amphipathic means that they exist, they can exist in both environments, basically. Like they have two regions, and one region is okay with the water and the other region is not okay with the water. So they're amphipathic. The hydrophobic region is these tails, and the hydrophilic region is the polar head group. So we're going to look at what that looks like. And you've probably heard of a phospholipid in biology. And phospholipids are incredibly important. Phospholipid. Because they inform so much about like our cell behavior. So phospholipids are amphipathic molecules that form lipid bilayer membranes, right? So let's look at what is actually a phospholipid. So we talked about the hydrophobic chains, which are right here, long carbon chains that don't like to interact with water. However, now over here, we have a polar head group. Hold on, I drew that quite badly. Hold on. A polar head group. And why is this polar? Because we've got this phosphate group with oxygen, and we've got some charge right here as well. So there's polarity here. This can stick up and interact well with the water. So these guys end up forming a lipid bilayer like this. So we can see, whoops, oh, I screwed that up. Oh, this is a nice picture too. Yeah, so the head groups that are interacting well with the water are up and below, right? So they're facing the water exterior to the cell, extracellular, and they're facing the aqueous solution intracellular, right? And in between, we have these hydrophobic tails that are happy because they're surrounded by other hydrophobic tails. There's no water in that region. There's nothing bothering them. So this is nice for them. They're happy with that. And so this is the basic structure of a phospholipid, right? And so, let's see. Yes, phospholipids. Okay, right. And so phospholipids, there's also sphingolipids. Sphingolipids are basically the same thing, but just a little bit different. So sphingolipids look like this. And so you can see here, if you compare the phospholipid and the sphingolipid, they actually have they're very similar in terms of properties, right? They have these long fatty acid chains, and at the top, they have something that is polar, that can interact well with water. However, this is a different molecule with different properties because it has a carbohydrate at the top, right? And carbohydrates are very, on a cell surface, are very important for signaling. So sphingosines are incredibly similar. You find them in the same place in the cell membrane, but have different properties. So it's good to be aware of them, right? And one big difference that could definitely come up, right? Notice how the phospholipid has two fatty acid chains, right? Because the glycerol backbone has three spots, right? Three areas where things could connect. One fatty acid, and then one to the polar head group. Look at the sphingosine backbone. This long carbon chain here, is not a fatty acid. It's not. Why? Because this is a fatty acid. Do you see that? This is not here. So that chain is not a fatty acid. So the big difference, phospholipids have two fatty acid chains, sphingolipids have one fatty acid chain. They look similar, and they function similarly, but there are some key differences. Okay, so now I think we're going to talk a little bit about membrane fluidity, right? So again, always we're talking about structure and we're talking about function. So Oh, degree of saturation. Yes. Okay. So saturation means when we're talking about saturation, we're talking about how much hydrogen is this carbon chain saturated in. So when we have a carbon chain, there's always these hidden hydrogens, right? There's two hidden hydrogens here at every carbon, right? Because each carbon is actually the end one is a CH3. But for the ones in the middle, right? This carbon is attached to a carbon, is attached to another carbon, and then is attached to two hydrogens, right? So this carbon, this hydrocarbon, the first one, is completely saturated because every spot that can have hydrogens it has hydrogens. However, what about here? What about here? The thing is, this carbon, right? This carbon right here is attached to a carbon, but then it's attached to that carbon again, right? And a carbon can only have 4 bonds. So it's got 1, 2, 3 bonds, and then it has 1 bond to a hydrogen, right? So this is considered a degree of unsaturation because it has 1 less hydrogen than it could have. Maximally, it could have up to 2 hydrogens if it's connected to 2 carbons. But here it's connected to 2 carbons, but it only has 1 hydrogen. So this is a degree of unsaturation. So why do we care about saturation? What is the point of saturation? When chains are fully saturated, they can pack together well because there's no kink. This bond, this thing here can cause a kink. So what does that look like? Here you can see that this left one is a long saturated fatty acid chain. And so they are all like relatively straight and they're all like able to pack together well. And between nonpolar molecules, there's van der Waals bonding. Like there's just like the small like transient interaction bond, right? However, what if we have a degree of unsaturation? This causes a bend because that bond is rigid, right? This causes a bend in the tail. When we have a bend in the tail, it's causing a gap and it's causing less binding between the phospholipids and it's causing less packing together, right? So this has some big effects on fluidity, on membrane fluidity. So what is membrane fluidity? Fluidity. So membrane fluidity is basically when we look at a membrane, it is not a static thing. There is something called the fluid mosaic model. And the fluid mosaic model is basically saying that everything in the membrane is moving around all the time, basically. Things are shifting up and down, left and right, and proteins can move around, basically. And for that to be the case, right, it's not like a static thing, but it's more like a sea. It's like a sea. It's like an ocean of these like phospholipids. And things can just move around generally in that ocean, right? However, just like an ocean has waves, right, depending on like the weather, membranes act differently depending on the temperature, depending on the environment, right? So membranes can either go how do I say, like membranes, the turbulence of a membrane will increase depending on the temperature or decrease depending on the temperature, right? So how does that work? Let's go back to phospholipids. How does that work? If the temperature increases, if the temperature of a membrane increases, or the environment increases, do molecules have more energy or less energy, right? This is a link to physics. This is a link to understanding why this happens, not just that it happens, but why does it happen? When temperature increases, kinetic energy increases. When kinetic energy increases, things are moving more, right? So if temperature increases, the fluidity of a membrane increases. And you know this intuitively, right? If your butter is melting, where do you put it? In the fridge, where the temperature is cold. If the temperature is cold, it decreases the movement of these lipids and it packs them together, right? So temperature increase leads to a fluidity increase. Now, when we're talking about fluidity, we're talking about a measure of how well they are packing together, right? If if something is so first let's talk about what if we decrease the temperature. If we decrease the temperature, then obviously the fluidity will decrease, right? Our butter won't melt. What is it? So the question now is, which structure is better at being less fluid? So when we say that we want to be less fluid, that means that we want to pack together tightly. When the phospholipids pack together tightly, then that's less fluidity because there's less gaps for them to move around. So if they're packing together tightly, should they be saturated and straight or unsaturated and kinked? Which one gives less space? If they're saturated or if they're unsaturated? If they're packed together tightly, right? Is there more space or is there less space compared to if they are kinked? Saturated has less space, right? So if they pack is so packing together tightly means that there's less space, means that they should be um unsaturated. Oh wait, saturated. Yes, sorry. Almost confused myself there. So the saturated phospholipids can pack together tightly because they don't have kinks, which decreases fluidity, which happens at low temperatures. So the reverse, what would increase fluidity? When they don't pack together tightly, in other words, when they're unsaturated and they have a kink, right? So we can write out and understand how these things relate to each other, right? Now, another factor that can affect fluidity is the length of the chain. The length of the chain. Let's draw this out to understand why a length of a chain matters. Let's say that this is just pretend like it's drawn well. Just pretend that this is a fatty acid chain and the carbonyl here is drawn properly. This is a fatty acid chain, right? If this binds to this, right, versus, so just imagine that these guys are packing together versus imagine this one. Which of these, the left one or the right one, which one do you think will pack together better? Which will have the more, so we can see from this picture, like the little red lines, right? The little red lines showing bonding, right? So which of these will have more bonding? Which of these will pack together better? If we draw out van der Waals bonds, right? The right. Why do you say the right? Right, it does, it has a shorter structure. Okay. So the thing is here, so you have a misconception, right? That a shorter structure would lead to more bonding, where it's actually the opposite. A longer structure leads to more bonding, right? So this is why we do this, so that you can, because there's a lot of things that you've picked up, or like maybe that you think makes sense, but is not actually correct. Like this also confused me so much when we talk when about like temperature and boiling and stuff. Like I had to like undo a lot of the things that I thought was correct but wasn't correct. In this case, when phospholipids are longer, there are more spots for them to do this like van der Waals bonding. So longer bonds pack together better, whereas longer chains pack together better, whereas shorter chains don't have that much carbon chains to actually bond with. So we can see that here. Here, the long one has all of this bonding, so it can pack together more strongly. The short one has less, it's missing this part. It's missing the rest of the chain. So then it loses all the bonding that it could have here. All the bonding that it could have here is gone because it doesn't have that chain. So the shorter chains actually will cause less bonding, less packing together. So how does that go back to our membrane fluidity discussion, right? If the shorter chains have less bonding to each other, then they are packing together less tightly. If they're packing together less tightly, then it's shorter chains. If they're packing together more tightly, then it's longer chains. So does that make sense? If it doesn't, tell me so that I can explain it in a different way. Okay. Okay, good. So now, yes. So this is so we spend a lot of time on this. Why? Because it actually we're not even finished talking about this, because there's other lipids that can impact the fluidity. So we're going to dive into that now. Let me see. Oh, I didn't even see this picture. Yeah, this is a nice picture talking about different phospholipid structures. You don't you I don't think you really need to know like the different types of head groups. You definitely don't need to know like the structure of a head group. You might need to like be able to recognize like, oh, a serine group or a choline group, like just the names that look like, oh, that looks like a polar head group. But the main thing is to understand, like, look at how these guys have 2 fatty acid chains and the sphingosine only has 1 fatty acid chain. Okay. So we talked about this, we talked about that, and we talked about that. Okay, good. So any questions overall on actually, no, wait, I need to talk about cholesterol. So we talked about unsaturation, right? So the double bonds introduce kinks. The kinks make them less likely to stack, less likely to pack together. So thus, room temperature unsaturated fatty acids are actually liquid. So oil has a lot of kinks in its lipid chains, and those kinks prevent it from packing together tightly. So therefore, at room temperature, it's liquid. Whereas like animal fat or other kinds of fat or like butter can be solid at room temperature because it is saturated fat. So it can pack together. It can pack together tightly. Okay. Glycerophospholipids, glycerol backbone that forms ester linkage to fatty acids and polar. Oh, glycerophospholipids instead of a single phosphate head group. Yeah, that's just these, right? Glycerol, right? They have glycerol in them, right? Then sphingolipids, we talked about the same thing, but they have one fatty acid tail. Triglycerides, we talked about, which are for energy storage, which they look like this, right? So again, the glycerol has three, sites that it can attach stuff to, right? Three sites that it can attach, right? In this case, on a fatty acid, on a triglyceride, the third site is just another fatty acid chain because the fatty acids are just being stored there to metabolize later for energy. Okay. So, and they're stored in specialized cells called adipocytes, or however you pronounce it. Okay. Where I wanted to talk about cholesterol. I guess I didn't mention it here, but that's important. Hold on. Hold on. Cholesterol, cholesterol, cholesterol. 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 Ah, yes, here we are. Here we go. So cholesterol adds another factor to membrane or fluidity. Cholesterol is like kind of like a bulky molecule in this membrane environment, right? So let me just clear up some of this. Okay, so cholesterol, what cholesterol does is it's kind of bulky and it's just in the membrane. So what cholesterol can do is it acts as a buffer. So what that means is it prevents either extreme. If it's extremely hot, the phospholipid bilayer is going to be very, very volatile and move around a lot. In that case, because the cholesterol is there, it is preventing, oh, this is showing without cholesterol. I need to show with cholesterol. Basically, at high temperatures, the cholesterol is going to be able to prevent the breakdown of the bilayer because it's just there and it's in the way. And so it's preventing the breakdown. So it will decrease fluidity at high temperatures. However, at low temperatures, right, it's very cold, and then the chains want to pack together. Because it's bulky, it prevents the complete packing of the chains. In that way, it increases fluidity at low temperatures. So notice that it has the opposite effect depending on which extreme that we're at. So that's why it's considered a fluidity buffer, because it prevents it going extreme. If we were at extremely low temperatures, then the membrane would pack together completely and would freeze, right? So it prevents that. It prevents the freezing by introducing a little bit of fluidity with its bulkiness. At high temperatures, if it was extremely high, then because they're moving around too much, the membrane might break apart, right? It prevents that by decreasing fluidity, preventing it from going to that extreme and dissolving the membrane. Okay, so that's just another aspect of what's important about cholesterol. Okay, so actually, I believe that is the major aspect of lipids. And let me see if there's anything about phospholipid, the bilayer that I want to talk about. I think the next thing we'll talk about is the cell membrane. No, this is viruses. Cell membrane. Okay, yes. So now we've basically understood like what's in the cell membrane from like its base components, right? We've got these phospholipids that make up the majority of like, the plasma membrane ocean. And then we have cholesterol, which is designed to stabilize the membrane. And you can find it in these things called lipid rafts. Not sure if I have a picture here, but basically the carbohydrates tend to like, not the carbohydrates, the cholesterols tend to be together in like a raft in the ocean. There are proteins there and there are carbohydrates there, right? And Yes. So before we move on, so now we're going to start talking about cells. Any questions about, these lipids and their functions and all that before we start talking about more about the membrane?
I actually have a quick question. Just to
Mhm.
confirm everything, I remember you mentioned something about the hydrolyzable lipids. Just like be on the same page, just for them to break down, there has to be a carbonyl group, correct? Like just present.
A carbonyl group attached that is attached to a leaving group. So that's a great question. So where was that picture? Where was that picture? What was that talking about? Maybe it was phospholipids? I'm not sure.
Yeah, we were talking about like the main topic was signaling lipids, and then
Oh,
you mentioned,
signaling. Okay, yeah,
yeah.
signal. Then maybe signaling. Uh, I know exactly what you're talking about, but I'm just trying to find that picture so I can show you the difference. Signaling. Prostaglandins, maybe? No, this is an example of the one that is not hydrolyzable because there is, but this is going to make more sense if I show you the other one first. Oh, yeah. Where is it? Where on earth is it? What card was it? I don't remember. Squalene lipids, something with lipids. Well, okay. In any case, let me just show you the structure of a regular triglyceride. Yeah, okay, so this is perfect. Okay, so this is a hydrolyzable lipid. Why? Because okay, I'm just going to clear off all this. Everybody got the stuff about fluidity and stuff, right? I'm going to clear this off.
Yeah, we got it.
Okay, so here, this is a good leaving group because that oxygen here, can just pick up a hydrogen. So that's okay. And then this guy oh, sorry, rather, yeah, this bond right here is where it's going to break. And so then this is going to become an OH, it's just going to become a regular glycerol. And then over here, because this is a hydration reaction, it's a molecule of H2O that's splitting that bond. So what's happening is that one H is going onto this oxygen, and then here I don't have space, so I'll just draw it like this. This carbon is then getting an OH. So that's hydrolyzable because this is now just a carboxylic acid, right? So, and then the other, so this looks messy. I'm just going to redraw it. So basically what you're getting is and then on the glycerol side, you just have the O now has an H on it. So this is hydrolyzable because what we added was right here. What we added was this H2O. Now, the reason that the other one is not hydrolyzable, okay, so do you see why that this is hydrolyzable?
Yeah, I can see that.
These are both okay. These are both like good leaving groups. I don't know how else, like these can exist properly and they're fine. They're stable rather. Now, if we look at the arachidonic, or if we look at the what is that thing called? Prostaglandin. If you look at the prostaglandin, right, we have a carbonyl here. It's not that we don't have a carbonyl here. The problem is, where can we do this kind of hydration reaction? We can't, because if we break this, right, and if we add an OH here, this carbon is not going to be able to, like, we can't do, it's not stable compared to if it was, if it if there was an O here. If there was an O here, then an O minus can exist. This is like organic chemistry, because if there's an oxygen, right? When we do this breakdown, the electrons here will go here, right? And when the electrons here go here, then this is temporarily an O minus. And that's okay. O minus is stable. It can just pick up an H. However, if we do try to do this here, then when the electrons come, it's going to be a carbon minus. That's not stable. Carbons don't like to be negatively charged, or carbons don't like to be charged in general. So that's why this doesn't really happen. So that's a little bit of organic chemistry. And this is a good question, because when you start doing the studying, you're going to see how all the different things that you've learned are all kind of going to start to blend together. And you're going to see like, oh, this concept that I learned in this class actually explains this concept in this class. Before I thought they were separate, but they are actually, like, structure informs function and function informs structure, and you'll start making those connections. So yes, good. Okay. Now what were we going to talk about? I think we were going to talk about cells or the membrane or something. All right. Any other questions? That was a good question.
I was going to say, thank you for the explanation. Really appreciate it.
Yeah, not a problem. That's what we do here. So now we want to talk about cell membrane, I believe. But where is the cell membrane? But I had a very specific card I wanted to talk about. Well, actually, let's just start here, actually. Oh, my cell. Oh, yeah, now I remember. Phospholipids. Yes. Sorry if I'm a little disorganized, but eventually I will find the things I want to talk about. All right, here we go. Okay. Oh, yeah. Okay. So another thing here, you can see that, well, actually, let's just read the explanation first, and then we'll see the picture. So we talked about the bilayers, right? And so we have this so the bilayers will make some cool shapes that minimize the free energy of the interactions. Again, we talked about that solvation shell. We talked about how nonpolar molecules are preventing the water from, are making the water a little bit more rigid, right? So there are some structures that they assume that maximize the water's capability to move, right? Even though they're nonpolar, they still have to exist in an aqueous environment of our body, right? So they will form these interesting structures like my cells. So my cell looks like this, and I think I have a better picture here. My cell looks like this, like a ball. And the way that the ball works is that all of the nonpolar stuff is facing inwards towards like the core, and all of the hydrophilic or the polar heads are facing outwards. So this is very efficient, right? All of the water is happy to interact with the hydrophilic heads, and all of the nonpolar hydrophobic tails are happy to interact with themselves on the inside. So this is what, and then here's like a cross section of what it looks like, right? All the nonpolar stuff is inside, all of the polar stuff is on the outside. That's a micelle. They are maximizing the free energy there. Now there's also bicells. They're just more, they look like a disc rather than a spear. They look like a flat, they kind of look like a red blood cell, if you know what I mean. And what else? And then the bilayer, right? So these guys are not layers. They are kind of just like they're kind of just like one layer, right? But then the bilayer, so we can see if I draw here, this is sort of like one layer, right? But a bilayer has two layers, right? So it's kind of even doubly effective, because now before, like for example, the water is out here and it's interacting fine with the head groups, and then inside is just nonpolar interactions, right? However, now we have nonpolar interactions between the layers, right? And then we have polar interactions, not only on the outside of the bilayer, but also on the inside of the bilayer, right? And guess what? This is a cell. This is what a cell is like, because inside of the cell is an aqueous environment. On the outside of the cell is an aqueous environment. So that's what a cell, that's why we have bilayer plasma membranes, because it's the most efficient way in terms of the free energy for the phospholipids to arrange themselves. So now we can talk about so also we talked about the fluid mosaic model, which is the fact that these guys move around, right? So for example, and they move around by themselves, right? Like spontaneously, but there are also enzymes that can make it happen a little faster. So movement left and right happens all the time. It's just water, right? It's just moving around like water. A flip base can flip it from the outer side to the inner side, and a flop base can flip it from the inner side to the outer side. Those are the names that are very aptly named: flip and flop. And you need ATP to do that. We talked about the fluid mosaic model. We talked about how carbohydrates, proteins, and less fluid stuff aggregate to create rafts that kind of float around on the surface of the membrane. We talked about cholesterol being in the cell membrane. We talked about signaling carbohydrates that are on the attached to the proteins on this extracellular surface that facilitate intracellular signaling, because carbohydrates can be recognized in terms of for signaling. And yeah, immune recognition, right? Because ABO blood grouping is based on sphingolipids, right? We showed you how the sphingolipid at the top can have a carbohydrate. Sphingo. Oh no, wait. Sphingo. Yeah, see right here how it has a carbohydrate at the top. So that can be used for signaling, and this will be embedded in the membrane. Okay. All right. So now we're going away from the lipids and just going to talk generally about what you can find in a cell membrane. So all right. So we talked about the phospholipids, we talked about the cholesterol, and now we got proteins. Now proteins are quite a large bunch of things that you can find in the cell membrane. And there's obviously, like, proteins have way different, like tons of different roles that they can play. Transmembrane proteins this is just in terms of structure, right? They are going throughout the whole phospholipid bilayer. And these guys are usually in terms of helping with transport, or they're receptors for signaling pathway, right? GPCRs are a type of transmembrane protein. We talked about GPCRs before, because they're like seven linked, 7 barrels that are transmembrane right here. 1, 2, 3, 4, 5, 6, 7. 7 transmembrane barrels. This is a very common signaling protein motif. Then you can also talk about what they do and how they do. Protein channels are basically kind of like tunnels that allow things to flow through, right? Channels, and a specific thing, when we're talking about channels, we're talking about passive transport, right? We'll talk more about that when we talk about like systems specifically. But the important thing is that, when things are passing through in and out of the cell, right? This is kind of like border security, right? We don't want just anything to come into the cell. We just want what we need to have to come into the cell. So what we need depends on what the cell needs, right? And it can change what it needs. So for example, if a channel is ligand gated, that means that something needs to attach to the channel for it to open, right? So that can be just like we'll talk more about that when we get to like action potential and stuff. You'll see that very commonly. Voltage gated ion channels, again, in action potentials, when the cell membrane reaches a certain voltage, it'll open automatically. And that's going to be, we're going to talk about that very in depth when we come to action potential. And then there are also membrane associated proteins, proteins that are not actually embedded within the membrane itself, but attached or associated with a protein. So let me see if I can find a good picture of that. So this right here is an integral protein because it is buried, it's integrated into the plasma membrane, right? But if there was another protein, you can also call that a transmembrane protein. However, if I drew like another protein over here, right, that just happens to be like associated with this protein, this guy right here is not an integral protein. He's a peripheral protein because he's not actually embedded in the plasma membrane. He's just there. So that's just a difference in terminology. Okay. Oh, okay. So we're going to talk about transport right here. Okay, perfect. So this will make a little bit more sense. Okay. This can get pretty involved, but we're not going to get super involved because this is more going to come up when we cover action potentials and the nervous system. So essentially, the main things are the fact that we have passive transport and active transport. Passive transport is flowing down the concentration gradient. So here you can see over here, right? These are small nonpolar molecules that can simply pass through the membrane and go from high concentration to low concentration. So if there's a bunch of them outside, then they're going to want to go to where they're not. That's just a concentration gradient. This is passive transport because it's not requiring energy. They're just going to do it on their own. Now, what if the molecules are polar and they cannot get through this, the nonpolar region of the membrane, right? If they can't, then there are these channels, right, that are going to allow them to go in. And we talked about how these channels can be closed, right, or they can always be open, right? There are different types of channels depending on how they work, or depending on the solute that's being transported. Again, this is not using energy because we're going from high to low concentration. So these guys can just flow down. So this is still passive transport. Now, active transport is the opposite. We're going from low to high. So we're going against concentration gradient. This low to high is kind of like a ball rolling down a hill. High to low is kind of like a ball rolling down a hill. That's how it's going to do it naturally. High to low, like a ball rolling down a hill. Low to high is like pushing a ball up a hill. So for low to high, we need to use energy to accomplish that, right? So what we do is, we'll talk about secondary later because it's not, or is it relevant here? Oh, it is relevant here. Okay, no, we will talk about it right now. So before we talk about that, basically, just to clear this up, simple diffusion, passive transport of molecule without a protein directly through the membrane, okay? Facilitated diffusion uses a transporter to move passively, right? Just because we use a protein does not mean that it's active. Using energy means that it's active. Facilitated transport is still going to be passive because we're going down the concentration gradient. Osmosis is water's movement from low solute to high solute, or in other words, high water concentration to low water concentration, right? And you probably heard of hypotonic, hypertonic, or isotonic conditions. That means like how the cell versus the environment. So hypotonic, isotonic. And you can see the behavior of the water. Yeah, this is fine. So if it's hypotonic, right? Well, first of all, isotonic means that the solute concentration outside and inside is the same. So there's no need for water to drastically move anywhere. Hypertonic means that the outside solution has way more stuff, way more solute than the inside. So if we just draw, let me just draw it so that it's clear. So let's say that there's 3 in here, and then there's 3 out there, right? Even Stevens, no, the water is not going to have any preference. Now let's say that there's only 1 here, and then there's 3 here, right? If that's the case, the water wants to go to where more solute is. So the water is going to, let me do it with blue. The water is going to go out to meet where the solute is. And that's when we have a hypertonic solution. My mnemonic for this back in the day was that if you drink a lot of sugar, it'll make you hyper, right? So just pretend like there's a lot of sugar out in the water. So if there's a lot of sugar out there, then if you drink it, it'll make you hyper. And then the opposite is if there's not that much outside, but there's a whole lot inside, right? If there's a whole lot inside, then the water is going to come rushing in and it's going to cause the cell to swell up. Like that. So these are the different types of tonicity. And this is based on osmosis, which is passive transport. It does it passively based on what is there. Okay. So now, oops, I gotta clear this all out. Osmosis. Then there's also aquaporins, which are again a facilitated transmembrane channels that allow water to come through in a facilitated way, right? Now active transport. So we talked about passive transport, which does not use energy. What is active transport? It uses energy, right? But there's 2 different ways. There's primary or direct active transport, which means we burn an ATP to move something to its high concentration. We burn the ATP and we directly move something. Secondary means that it's actually interesting. Secondary transports are like so the primary transport is like the sodium potassium pump, right? Where we move sodium and potassium using the ATP. And oh no, we don't need this right now. So then secondary active transport secondary active transport processes power the unfavorable movement of a solute, like an ion, against its concentration gradient by simultaneously moving another solute favorably down the gradient. So what does that mean? If we have the sodium, right, it's going down its gradient and it's going down the gradient, right? There's more sodium here and there's less sodium here. So the sodium is going where it wants to go. However, at the same time, we'll take whatever solute we want with it. I think it's glucose. The glucose will take with it into the high concentration zone. So the only reason this works is because we use some ATP to in the first place. And so the energy that releases by this going down is also allowing us to take the solute where it doesn't necessarily want to go down by its own. So these are both using energy. And there are different ways that we can see that. So what we saw here is a symport because both the sodium and the solute are going to the same place. On reverse, you can have an antiport where something is going one way and something else is going the other way. So these are 2 different types of active transport that we can see. Let me see if active transport. Oh yeah, here's another picture. Yeah. So we see here we did primary active transport by burning the ATP to move the Na up here, to move the Na where there's already plenty of Na, right? So we moved it. Now, once we move the Na to the high concentration zone, the Na is naturally going to want to come back down to the low concentration zone, right? That's what this yellow arrow is showing here. So when the Na naturally comes down, right, we spent some ATP to already move it up, and now when it comes down, it's going to bring the glucose with it. So that's why this is still active transport. The secondary is still active transport because this only happened because we used ATP in the first place. So energy released by the sodium traveling down its concentration gradient was used to transfer glucose against its concentration gradient. That's our two types of active transport. Okie dokes. Okay. So that's about it for transporters. Okay, no worries. Have a good weekend. That's about it for transporters. And let me see. So the next part was going to be about membranes. How much time do we have? We have time, yes. Okay.